Hello and welcome back everyone to the video series in which I am talking about the Lac Operon, that is a gene expression in prokaryotes. In this particular section of video, I am going to discuss about the Lac Operon and the concept of and working of the Lac Operon. First of all, before talking about Lac Operon, I want to tell about what is gene expression. Alright, whenever the information from the gene is passed to the protein and the protein shows the functional activity, finally that functional activity is known as gene expression. For an example, we have a gene. Gene is a hereditary unit, right? DNA is a hereditary unit, and why is it called hereditary unit? Because there is certain kind of information, and this information can be like, we can get this information finally, this information can be functional finally when the gene will express. And when will the gene express? The gene will express when what happens? It will form mRNA and then protein and then finally the expression. And why we study this particular operon for? To study or to know the gene expression, because within this single set of gene, we can see all these four processes going on. That is the gene. This is a gene. And then mRNA, the one it will transcribe. This is mRNA and this will also give mRNA. And the protein, these all are the protein produced by the mRNA of this gene. Alright, and the expression, we will see the metabolism of the lactose. Alright, within this one particular operon concept, we will be understanding all the four things, that is the gene, it giving mRNA and the protein and the expression. Alright, before the discovery of this, or our study of the gene expression was only virtual, it was not so real. But when we came to know about this, then our finding, then we came to know this, yes, with the finding which we made, that is transcription, translation, all those things are real, and we can describe it through one example, that is operon concept. Now, first we will be talking about the initial stage. Alright, normally in a bacterial cell, this particular operon is always in off mode. Alright, in normal, this operon is in off condition. Only when the bacteria will not find a source of glucose, then what will it do is, to produce energy, it will utilize lactose. Alright, these will be on only when there is no glucose in the cell. Alright, and then by default this is off, because see here, this PI means Lac I promoter, and Lac I means this is a gene which is responsible for the formation of the repressor protein. This gene transcribes repressor mRNA, and this repressor mRNA, it gives repressor protein. Now, this repressor protein will go and bind onto the operator. When the repressor protein has bonded to the operator, then what happens is the transcription of the gene which codes for the enzyme will not occur, because this operon is off. When the repressor will bind to the operator, the operon system will be off. Only when the repressor binding is not there in the operator and this is free, then only what happens, RNA polymerase can sit in the promoter and can transcribe the mRNA from this particular gene. So first, this is in a off mode now. Now, for instance, what happens is there is no glucose in the cell. This is a cell, for example, this particular thing is a cell membrane, for example. Now there is no glucose inside the cell. So what happens is the enzyme beta galactosidase, galactoside permease, galactoside transacetylase, these are the enzyme. This is produced by Lac Z, this is produced by Lac Y, and this is produced by Lac A gene, which is over here. Now these enzymes will be inside the cell. Alright, these enzymes will be by default or normally be present inside the cell, but in very less number. Alright, from the previous cycle of the Lac Operon might be, or sometime the repressor might fall, and then RNA polymerase can synthesize a small stretch of this enzyme. So few, like 5 copies of each enzymes are present inside the cell by default. It is because since these enzymes are required by the cell to on the Lac Operon, so it is obvious that some few copies might be there inside the cell to utilize whenever the lactose comes, right? So what happens is, when there is no glucose and there is lactose in the medium, then these 3 enzymes get activated. When these 3 enzymes get activated, what they will do is, this permease enzyme is a enzyme which will embed in the wall of the cell membrane. The permease enzyme will get embedded in the wall of cell membrane. Only 5 copies of each enzymes will be there, not much. And then what happens is, there will be the lactose in the medium. Outside the cell there will be a lactose. That is why this operon is on. So lactose will enter the cell. Alright, when the lactose will enter the cell, then this beta galactosidase will break the lactose, this lactose will be converted into its isomeric form, that is allolactose. Alright, these will be converted, these lactose molecule, some of the lactose molecule will be changed structurally into allolactose, that is the isomeric form of lactose. Now these allolactose will go and bind to the repressor protein. When this allolactose will go and bind to the repressor protein, what it does is, it does a conformational change of the repressor protein. The conformation of the repressor protein will be changed. So now this repressor protein will, this reaction is suppose going on here, it is binding over here. Now its conformation will be changed. So now the operator site will be empty. That means when the operator site will be empty, then the operator is on. Whenever the repressor protein is bonded to the operator site, the Lac Operon is off. Now when there is conformational change in the repressor, this operator will be on now. Now since the operator is on, what happens is, RNA polymerase will come and sit onto the promoter region. Since RNA polymerase is long, it will cover certain distance. And then what is there is, near the operator, exactly in the upstream near the operator, there is a site in which there is a site for the bonding of CAP, that is the catabolic activator protein. This is the site in which the catabolic activator protein can bind. Catabolic activator protein is a activator protein which helps in the process of transcription of mRNA. While studying about the transcription, we study about the different type of protein, right? Different type of activator protein which is employed during the process of transcription. This CAP is also one of the activator protein which binds near the promoter because it has a site. It has a specific site to bind near the promoter. Now, when the catabolic protein, when this cap binding protein will bind to CAMP, cyclic AMP. For example, this is cyclic AMP and this is CAMP and this is CAP. This is catabolic activator protein and this is cyclic AMP. They will form a complex. So first let me tell you, when there is glucose plus lactose, when there is both glucose plus lactose in the medium, then the Lac Operon will be off. At this time the concentration of CAMP will be very much low. When the glucose is present, the concentration of CAMP, cyclic AMP, will be low. But when there is no glucose in the medium and only lactose in the medium, then the Lac Operon will be on and the concentration of CAMP will be very much high. Alright, it is transversely proportional. When the glucose is present, CAMP is less in concentration, and when the glucose is absent, the CAMP is more in concentration, and when the glucose is present, CAMP is less in concentration. And here in this we are talking about this condition. We are not talking about this condition now. We are talking about this condition because we are studying about the Lac Operon, and Lac Operon will be on only when the lactose is there in the medium and there is the glucose absent in the medium and the concentration of CAMP is high. Now, when the concentration of CAMP is high, cyclic AMP is high, then it will bind with the catabolic activator protein, and this complex will bind to the CAP binding site. This is known as CAP binding site. Now, these will bind to the CAP binding site. This particular complex will bind to the CAP binding site, and when this will bind to the CAP binding site and allolactose bind to the repressor, and when the RNA polymerase 2 this is only RNA polymerase because it is for bacteria, and when the RNA polymerase will bind to the promoter and cyclic AMP plus cap complex will bind to the cap binding site, and when the allolactose will bind to the repressor, now finally what happens is transcription of the mRNA from this gene will begin, and when the transcription of this gene will begin, then they will synthesize beta galactosidase, Y will synthesize galactoside permease, and E will synthesize galactoside transacetylase in a very huge copy numbers. Alright, before only there was few 5 molecule of each of this enzyme inside the cell, and whenever the cell found that there is no glucose but we have lactose, so we have to utilize it, so it gets activated and it makes a permeability to the, it makes the lactose permeable to the cell, and they will convert, the lactose will be converted to allolactose and it will bind to the repressor, and when the lactose will bind to the repressor, there will be the conformational change and the operator region will be vacant and the promoter will go and bind. At this condition, CAMP plus cyclic AMP complex will also bind to the cap binding region and activate the process. Now our mRNA is formed and all the genes are formed, so there will be frequent entry of the lactose in a large number. Alright, there will be the frequent entry of the lactose because now permease is also formed in few more than 5,000 copies. Now more and more more access to the lactose. There is more and more channel for the entrance of the lactose now. Alright, more and more channel for the entrance of the lactose. So lactose will be able to enter from all the places from here and here and here, and there will be the large number of lactose. So when there is large number of lactose, beta galactosidase will break it into glucose plus galactose, and it will be like permease will enable the more and more entry of lactose. Beta galactosidase will break more and more number of lactose. But what happens is the availability of lactose is so much, right? So there must be someone to help beta galactosidase, right? So this galactoside transacetylase, it acts as a helper hand. What happens is it transfer the coenzyme A. Alright, transacetylase transfer the coenzyme A from the acetyl coenzyme A. It will transfer coenzyme A to the beta galactosidase and help the beta galactosidase to do its work more frequently, to break the glucose and to break the lactose into glucose and galactose more frequently. Alright, galactoside transacetylase will transfer acetyl coenzyme A to the beta galactosidase and help it to break the lactose into glucose and galactose more frequently. Now what happens is all the lactose has been utilized now. Alright, so what will happen is after utilization of all the lactose present in the repressor, the isomeric form of lactose, that is allolactose present in the repressor, will also be utilized now. This is also one form of lactose itself. So after the utilization of all the lactose, this lactose at the end will be utilized. Now what happens after utilization of this lactose? Our repressor will be free again. Alright, let me draw it here now. Alright, now our repressor will be free again, right? This will be utilized and the repressor will be free again. So now it will be coming back to its original conformation. Alright, when the last lactose is also utilized, our repressor will be free and it will be coming back to its original state again. So now when it will be coming back to its original conformation, now it will again go and bind to the operator. So now what happens? RNA polymerase will fall off and the transcription will stop, and these complex will also be discarded. Alright, because now due to the breaking of the lactose, glucose is produced in large number. So when the glucose is in large number, then what happens? The concentration of CAMP will decrease. So this complex will also break and the transcription and translation and the metabolism of lactose will finally stop as the operon stops. Alright, this was only the Lac Operon concept, and I hope you got the thing, and I try to accommodate all the things in one video and make it short and cute. If you like the video, please hit the like button and subscribe to my channel.